Hello, everybody. So it's my pleasure to be here today and to. Well, the idea is to share with you some idea about sequencing and perhaps what can be the implication for our specific project on DNA storage. And I thought that it would be interesting for some of you to come back to the old history and how just the notion that DNA is playing a role in biology came out, and what are the implications that it can have for our projects. Okay. The first thing that is remarkable is that the nucleic acid sequencing by itself is a very recent technology. So we have not even celebrated the 50 years of the discovery of sequencing. It was a paper by Sanger in '77. And so it has evolved very rapidly and to what we are knowing today. And what is interesting is that it informs us, the technology itself informs us about two major biomolecules that are shared by all life forms on Earth, the RNA and the DNA. And the use of the sequencing has transformed completely the analysis of life and the different forms of life. So the idea is to think about the succession of technological breakthrough that move from a very expensive and tedious and difficult technology to something that represents nothing today and that is very affordable and that can provide at a very cheap price terabytes of nucleic acid. So that also implies, and this is exactly the essence of MoleculArXiv and the type of project behind, that such progress are also opening a lot of new fields of applications. And among them, there is naturally the DNA storage of information. So I will probably say a lot of evidence, but it's always very important to remember, for instance, the very high-speed progress during the last 20 years, with a doubling of the capacity of sequencing every 5 months at the beginning of the 2000s. And the development of an ever-growing range of experience and needs. So that led in France to the creation of what we call a national infrastructure, which is called France Génomique. And so my lab belongs to France Génomique, and this is a series of platforms that are located all over France, and the general idea at its creation was to maintain competitiveness of the academic laboratories and to offer the highest range of expertise and skills and the capacity to support ambitious projects. So the infrastructure itself is located all over France, and you have the different spots. And our platform is located in Sophia Antipolis, and it is called UCA Genomics. So just to give you a flavor of the kind of things that we are doing, because we are not only sequencing the shells for MoleculArXiv, but we are, for instance, involved in the Human Cell Atlas project, and we have contributed to the elaboration to the first version of the Human Lung Cell Atlas. And in the context of this project, we have also developed some new technologies, for instance, to combine single cells and long-read sequencing using nanopore sequencing, and also using the nanopore sequencing in the context of spatial isoform transcriptomics. And that was some of the recent outputs that we have made in that context. So I can skip to the other examples, but sequencing is everywhere. And so, for instance, if you consider what came out after the pandemics, it turned out that it was very useful to monitor what was going on in the wastewater of the big cities, because it was a direct indicator of what was going on in terms of the viral loads for different type of virus species. And for instance, here you have a 4-year survey of the different types of the different variants of SARS-CoV-2 that came out in the wastewater from Nice. And it is interesting to compare that picture with the pictures that you can get with clinical samples. And what is amazing is that with one sample every week gives you as much, if not better, information than thousands or tens of thousands of clinical samples that you have a lot of difficulty to collect and that are very expensive to sequence. So there are at the moment some efforts to widen the picture and apply this type of technology not only to SARS-CoV-2 but to a lot of different viruses. You can consider all other respiratory viruses. You can also consider intestinal viruses, or for instance, the influenza for the birds, the avian influenza, which is also a concern. But going back to the plan, my first question will be why DNA, and how do we demonstrate that this is DNA that we have to follow, and from where is it coming? And for that, I think that it is interesting to come back to the concept of heredity and how it shifted, the question shifted from the question of heredity to the question of handling of information. And then I think that the next level is to understand the different type of containers, of biological containers that store information. You have very diverse form of storing of information storage in nucleic acids. And this is exemplified by the situation in different viruses, in bacteria, in multicellular eukaryotes. And so in the last part of my presentation, it will be more focused on the technology of nucleic acid sequencing by itself. So the why DNA has already been discussed yesterday and introduced by Marc. It is the best example that we can have is coming from, for instance, the work by Svante Pääbo with sequencing of all species like the Neanderthal genome. And it's interesting because it gives you a flavor of what was going on 100,000 years ago. And you can compare directly the sequence of modern DNA by comparison with an ancient DNA. So this is interesting. You have a first observation to make because you see that the sequences are longer with the modern DNA, just because you have cleavage, chemical cleavage in ancient DNA that takes place. What is particularly important to notice is that the remains that have been used when you consider, for instance, the Denisova man that has been sequenced by Svante Pääbo and his groups, was stored in a cave, so it was in high altitude, the temperature was low, but there was no specific attention paid to the level of water, so they didn't pay attention to the imaging capsule to store their DNA, but nevertheless it was possible to elaborate a quite impressive sequencing. And so I will share with you some observations that I made when Svante Pääbo got the Nobel Prize. So I was pleased because it was a very nice Nobel Prize. But, and so I went to his website, and they have stored all the information about the different genomes that they have sequenced. And so I align the Denisova man, and I look at my favorite gene. I have worked on cystic fibrosis, so I look on CFTR, which is the genes for the cystic fibrosis. And I made a major observation, which is the fact that there was a mutation there. And there was an heterozygote mutation. So that I was very excited, and I saw that it was a big discovery. And there was two things that were amazing. It was putting the history of cystic fibrosis 50,000 years back in the history, because it was considered that it emerged in the 15,000s before Christ. And here we were working with a sample having 100,000 years. But there was something that was weird, and I discussed with specialists of CF genetics, and the first thing is that when you align that with modern DNA from heterozygotes, you see that there is a shift. And by the way, the mutation corresponds to a TTT in the Denisova man, while it is a CTT in the modern DNA. So there was a second discovery that was perhaps the bad one, which corresponds to the fact that there was a different mutation. It was not the founding mutation that has been found later on, and that corresponds to the most frequent mutation of cystic fibrosis. At the end of the story, I have discussed with many geneticists and specialists of cystic fibrosis, and we were not really convinced by the nature of the data, and this is why I wanted to share it with you today, just because you can store information, but you have to be very cautious about the nature of the signals that you will be detecting. And in that case, what happened is that it turned out that we found that only in one specimen, but the specimen of Neanderthal are rare, and Denisova are even rarer. And when we looked at this mutation, it was in a place where we have a very weird coverage with a small peak of reads. And it was only in one direction, but we were never able to get the reverse sequence. And so that, and on top of that, we have screened a lot of population, of human population, and we were expecting that because there are some transfer of information between the Denisovan and the modern population, there is also a transfer from Neanderthal to the modern population. We looked in modern population if we were able to detect this particular polymorphism, and it was never identified. So we decided that it was a fake mutation, but that's important because it implies that when we are storing information, we can be facing this type of situation, and this is something that we have to consider in our project. But so after this first and negative results, let's come back to the what is probably for biologists the ultimate question of life, universe and everything. The expression in English is not the good one. It is better to consider the French traduction. Les chiens ne font pas des chats. Dogs are not making cats. And this is illustrated on the left part. And behind that, the big question is about the question of heredity. And you have another illustration with all these pictures of the Habsburg family with, with these very characteristic pictures that suggest that something that everybody believes. Heredity is recognized. It is recognized forever. But it was not explained scientifically. And so the first question was about, much before the idea of the gene concept, was how to explain these heredity traits and how can we understand how they are transmitted from the father and mother to the children. And so that's interesting because you will see that there are some theories that are back and forth. They are forgotten and they come back in another form. And first illustration is given by the pangenesis theory from Hippocrates, so during the Greek time. And he suggested that pangenes were tiny particles from all the parts of the body that were collected in the reproductive organs and then passed to the offspring. So that was the first idea to the transmission of genetic traits. Then Aristotle came, and I'm sorry for the woman, but the idea was slightly different. Both parents were considered to have an influence on the offspring. The male were providing the form and the cause of the movement. And during that time, the female were providing just the matter and the substance to be shaped, resulting in a blending of the parental contribution. I'm sorry, but this is Aristotle. And again, it's interesting because, well, the theory will come back and you will see later on. We have to come to the 17th, 18th century to see that the notion to mix the parental traits are evolving, but there is still no notion of the discrete heredity units that will correspond to a gene by itself. And the first notion of that is again, well, the pre-formationists like Leeuwenhoek, one of the creators of the microscopes, and Malebranche believe that the embryos are already existing in miniature form and that they are just unfolding when they develop. And a slightly different view was taken by Wolff, who pointed out the progressive development of organisms, and that was the start of the developmental biology, understanding that you can come from an undifferentiated state to a differentiated state, which is what we believe now. And then there was a very important time with three French biologists, Cuvier, Geoffroy Saint-Hilaire, and Lamarck. Cuvier and Geoffroy Saint-Hilaire was particularly interested in what they have created, the paleontology and the study of the remains. And what was important then was not that they didn't work directly on the heredity, but they found that there was some internal structure in a whole organism that can be characterized by a functional link between the different organs. And so the preservation of forms is reinforcing the functional integrity. And what is interesting is that in parallel, Geoffroy Saint-Hilaire created a theory about the unity of plan. And what is interesting is that the two sides are related. But in one part, you will consider that heredity will maintain stable and integrated form. So you will create an organism, another organism, and the co will not be, will have no link with a dog or a human or a mouse. While with Geoffroy Saint-Hilaire, you have the unity of plan that creates something that can be a modifiable plan of organization. And so you are coming with the first notion that you can have an evolution that can link together all the different forms of life in something more common. And Lamarck arrived at nearly the same time with the idea that the function creates the organ. So the idea that the giraffe will go at the top of the tree and will develop its neck. And this is the first explicit mechanisms of evolutionary change that has been evoked. But naturally, the big man in that is Darwin, with a book in 1859 about natural selection. And the work by Darwin is creating a new concept about the way that there is an advantage in the population and in a specific context for some individual that will be favored and that will develop. The problem is that in his first book, he was not defining the model of heredity itself. And so we don't know at that time anything about the mechanism, the mechanistic part that will lead to the creation of the transmission of heredity factors. And so what is interesting is that he's coming back to Hippocrates, and he suggests aggregation of reproductive organ of gemmules that are released from all the body cells. So this sort of migration, which is not very well recognized, but in a way you can also interpret that in a different manner and consider that he's mentioning what if you replace the gemmule by a gene, you can be right, and that can correspond to the right theory, but I'm not sure that it was exactly formulated like that. And well, he was needing some additional information that came later on, and this is Mendel that provides them. But the work from Mendel was totally ignored till the beginning of the 20th century. But working on peas, he was able to establish mechanical rules that govern the transmission of characteristics between different varieties. And he invented a law of segregation between parental factors that are separated in gametes and the law of independent assortment, and making the notion that there can be some discrete hereditary factors. And in fact, it was the genes that were governing the inheritance. So he was not alone to have made similar observation. Were also developed by Naudin and Gartner, but the credit is totally to Mendel, who did that in 1865. So the notion was that the traits do not mix but are transmitted in stable units. And then, in parallel, there were a lot of progress that were made in the biochemistry and the biochemistry. So, for instance, with the isolation of the first DNA from salmon sperm by Mischer, then the identification by Nägeli of two types of substance in a cell, and we are coming back to Aristotle with the notion of the trophoplasm, which is responsible for the nutrition and the growth, and the idioplasm, which is located into the nucleus. and the idioplasm is the basis of heredity and transmit characteristic to the descent. So we are at that time already elaborating a lot of things that comes in the direction of what we know today, with this notion of nucleus that contains important information. The link with, between that and DNA is not totally done, but it is near. And Weissman is then coming around that, and he reinforces the different works by distinguishing the germplasm, which is transmitted to the descendants, and the soma, so the body, which is not hereditary. And after that, the rediscovery at the beginning of the 19th century of the Mendel's law led to what they call a pangenesis, to define the units of heredity. And this will be, the theory will be completely stabilized by the work by Morgan in the fruit fly, Drosophila melanogaster, where with his colleagues he was able to physically identify the genes on different parts of chromosomes, and by making precise location of genes on specific chromosome, and by mapping their distance by measuring the frequency of recombination between events, they were able to establish strongly the notion of gene, and that was a very, very strong demonstration. So in parallel, Avery, MacLeod, and McCarthy made an experiment in Streptococcus pneumoniae that DNA rather than proteins was the transforming factor that was converting a bacterium into a non-virulent bacteria into a virulent one. So that was at that time an experiment where they mixed different types of radioactivity, one that was associated with nucleic acids, the other to the proteins. They were following this radioactivity, and they were clearly associating the transmission of a character to the transmission of DNA and not to the transmission of proteins. And so everything was set, and it is at that time that Schrödinger in 1944, introduced the concept of heredity as information storage. And so he conceptualized the notion of an aperiodic crystal that inspires then Watson and Crick for their work on the structure of DNA. And so now we are arrived, and we are with the notion that the DNA carries the genetic information. The demonstration has been made in different species. So formally, you can say that it has not been demonstrated in all the species, but you have a strong pressure to indicate that it is probably true to all of them. And the gene has become a material fragment that contains information. So this picture is a beautiful picture. It was taken by Rosalind Franklin, and then it inspired Watson and Crick, who were crystallographers and who were able to conceptualize the double helix from this first information. It is amazing how a small load of information, less than 30 kilooctets, has given such an important information, but this is the case. And from that, it was possible to elaborate more precise theory at the same period. The same people, so Crick, Watson, and many others, were aware of the great importance of two different types of molecules. So we discussed a lot already of DNA, and it was nicely introduced yesterday. So we know exactly the structure, the combination between A and T, G and C. We know also that there is an equivalent under the form of RNA, where the desoxyribose is replaced by a ribose, and in that case, the T is replaced by a U. It is important to notice that this existence of RNA is not only limited into our cells to the transcripts that are the vector to go from gene to proteins, but it also represents the genome itself in the situation of many viruses. And so you have different forms of genomes that are recognized, from single-strand viruses, double-stranded, you have linear or circular genomes. And so you have a large variety of them, and we will discuss that in more details. What is interesting is that regarding the type of coding, so I know that you will be discussing a lot of very elaborate code and the way to be very efficient and to get the most information of it. What I can share today is what has been selected by the nature in the case of the living systems, and in that case, the coding is a direct coding where codons, represented by 3 bases, are transferring information via a shuttle, which is called the transfer RNA, into protein, and you have the synthesis of protein that can grow and the peptide chains that can extend. So in that case, and in a way, this is the model that you have used, Dominique. We have a simple coding, but which directly link the sequence into something that can be decrypted quite directly. So when you consider the different transfer RNA, so if you have 4 power 3, you have 64 combinations, and this 64 combination gave to the notion of a genetic code. Usually it is considered as a universal code. What has been written by Crick when he wrote the first paper was the code is universal or nearly so, and I insist on the nearly so, because if you are looking now at different species, so just for instance, if you compare the eukaryote, the standard code and the mitochondrion genome, you have slight difference that correspond to some codons that becomes stop codon in the mitochondria that are not in the universal code. And you have other instances where you have different codes that are associated with different species. So this is the right mention is a nearly so from Francis Crick. But what is interesting is that this was the capacity for a triplet code to pass on amino acid. And so that was nicely demonstrated at an heroic period where it was so difficult. At that time, it was not easy to synthesize oligo. And so what people did first was that they synthesized poly-U, poly-A, poly-C, and they discovered that in that case they were able to translate a polyphenylalanine, a polylysine, or a polyproline. And that was the start of the elucidation of the genetic code. And then by different trials, it was possible to elaborate the full story. there is also some different initial triplet of nucleotides and stop codon, and that change, in that case, the possibility to define what we call an open reading frame that corresponds to the full sequence of a protein. So at that point, we have a genome, so the entire genetic material of an organism. I said that it was not necessarily DNA because it was possibly also RNA in the case of some viruses. And so all this information is able to code protein coding genes, structural RNAs. I mentioned the transfer RNA that are necessary for the translation. But you have also ribosomal RNA, you have a lot of different RNAs that have some functional role into a cell, and that probably refer to, well, at least this is a hypothesis. The hypothesis is that in the history of life, the first form of life were probably driven by RNA, and then this RNA was able to have some enzymatic activity, and it evolved with the creation of protein and then and DNA. So the structuration that we will detail now is about the subdivision of the genomes. This can be in different chromosomes that consist in a single DNA molecules. You can have linear molecules in eukaryotes. Very often they are circular in prokaryotes. And there is also a very unique genome that is found in our cells that corresponds to the mitochondrial genome. And this is unique because it comes probably from a symbiotic mechanisms that took place long time ago and that resulted into the entry of bacteria into an eukaryotic cell. And they make these two cells made a deal and were able to work together. The mitochondria, or what became the mitochondria, was generating most of the energy needed by the cell, and the cell was taking part of the genome to get all the information. And so there is a very strong reduction of the mitochondrial genomes. And for instance, in the human genome, that represents only 15 kilobases, which is very small by comparison to the 3 gigabase of the full human genome. But each cell can contain hundreds or thousands copies of circular DNA molecules. And so you have a lot of information from the mitochondria, which is in fact floating into the cells, and that will have an impact on all the measurements that we are doing. Okay, so the different sequencing, I can go very fast, but when you have the information coming from many different genomes that have been sequenced, immediately you can compare the sequence. You are able to make relationship between and distance between the different members. You can draw this map of life and elaborate what are the different branch of the tree of life. And in that context, so we have different situations, as I mentioned. The first situation, and I think that this is also interesting for you to understand that, because there are some beautiful instruments to carry information from one situation to the other. So viruses can be very potent to transfer from one spot to the other. But we will see that they have a very compact genome and they are not necessarily able to transfer a lot of extra information. So meaning that we can perhaps take some inspiration about the mechanisms that they are using to manipulate the information. But I'm not sure that they will have all the capacity to transfer big libraries of data from one place to the other. That can be discussed perhaps at the end of the presentation. So another aspect is given by the bacteria. So they represent what we can say a minimal cell. They are very efficient, and they have also very compact genomes with, that can be driven, for instance, by operon. And then we arrive to very complex eukaryotes. And so I will just detail very rapidly different instances to see how this can draw a different strategy for long-term storage of information. So one thing is that you have the DNA as a physical object, and you will put it into different receptacles. So that can be a capsid in the case of a virus. You have the bacteria, and you have the eukaryotic cells. And for that, you need some packaging, because we will see the size of the genome can be quite significant. And so you have different mechanisms that are at work to allow the packaging and that are quite different. With, for instance, in the case of the viruses, an extreme confinement and the physical control that makes the virus able to jump into the cells because when the capsid opens, there is such a pressure that it will be released very fastly into the infected cells. While in the eukaryote, you have something that is taking place at an equilibrium of pressure, and there are additional mechanisms that are necessary to allow the good organization of the genome. And so this is what we call the nucleosome condensation. Yeah, so I will not spend too much time on the different classification of all the viruses, bacteria, etc. I can perhaps just give a brief example. Well, in biology, nearly everything is possible. So you will have, for instance, double-stranded DNA viruses. You will have single-stranded DNA viruses. You will have double-stranded RNA. You will have single-stranded RNA viruses that are directly translatable. This is the case, for instance, of the SARS-CoV-2. You have also the single RNA sans minus viruses. For instance, the respiratory syncytial virus. And so all the combinations are possible. And they are driven by a very specific system. But in all the cases, what happens is that you have the capacity to accomplish multiple tasks with minimal genetic information. You have to recognize that the size of a genome, like the respiratory syncytial virus, is about 10 kilobases. So it's a very short and very compacted. And so, for instance, it is the same polymerase that will do at the same time the replication to generate a new copy of the RNA for the next copy of virus that will be generated, and the transcription that will generate all the RNAs that are necessary to translate translate into proteins and to generate all the molecules that are necessary for to elaborate a fully operational virus. And so this is controlled by very specific system. And so, for instance, the difference between replication and transcription is related to an initiation at a position +1 in the sequence, while the transcription will take place after an initiation at +3. So you just shift a little bit the position at which you start, and in that case, you have the translation of the transcriptions that are taking place. And so you see the exquisite sensitivity and the efficiency of the system in such a case. So another system that I wanted to mention is the operon, which is a specificity of bacteria, in which this feature of prokaryotes makes that a single promoters can control several genes that are organized in functional units. So there is nothing really similar or exactly similar in eukaryotes. There are some exceptions, but for instance, the very famous one is the lac operon that was identified by Lvov, Monod, and Jacob in the 60s. And this operon provide efficiency, rapid adaptation, and so they can co-express all the molecules that are necessary for a system to work. I can skip because I already mentioned that. And now I wanted to describe a little bit the diversity of the genome size. Again, I will not discuss everything, but I will just compare the size of the genome in plants between a very compact genome like Arabidopsis that you have here, that is made of the nuclear genome correspond to 130 bases, and you have on the right the Paris Japonica, which represents a genome which is 1,000-fold larger. So I'm not sure that this is what can be so different between the two organisms to generate this increase, but there are some outstanding differences, and we will elaborate a little bit more about that. So I already mentioned the fact that the way that the size of the genome was elaborated it can be very simple. I mean, when no sequencing was possible, if you were able to isolate a cell from human, for instance, if you can know exactly the number of cells that you have in your sample, if you extract the DNA and you get the genomic DNA, you weight the quantity of DNA that you have purified, and you have directly an indication about the size of the genome. So when we had no sequencing, this is how we were detecting the size of the genome, and we were already able to get some information. So when we do that, the human genome, probably 1 or 2 meters per cell, and you can compare it with the other elements. So this is a viral. This is a virus that infects the bacteria. This is a bacteria. This is a human, and you have different order that makes the different characteristic of the genome. So yes. So the next thing is to know how you organize the information. So in the case of the human genome, well represented here, we know that we have 23 pairs of chromosomes, but you have also, despite the fact that you have the same kind of function between human, chimpanzee, or even dog, mouse, etc., you see that you can recognize a very large diversity. So sometimes it has been perhaps affected by the growing and the identification of specific species, but there are some plants. So, for instance, this is a fern where it was identified more than 1,000 chromosomes. And by the way, they were identified, and the firm is able to go into mitosis, and it's a mess. It's a total mess. But however, it is working. And so you can raise to this huge number of chromosomes, and you are still able to maintain a viable species. So that's something interesting, and well, in a way, that gives us also the idea that perhaps a sort of mini chromosome can be a good way to or a good consideration for a mechanism of storage, and we are not necessarily limited to the number of chromosomes that we are used to know. For instance, the 23 that are known in human. But so the next thing that I wanted to share there was the organization, the more detailed organization, because when we are saying that we have 3 gigabases of data over 22 autosome two sexual chromosomes and 22 autosomes. In fact, what happens is that a lot of this information is not coding at all something that is directly involved into the function. You have a lot of information that correspond to non-coding information. This is even more than the 50% that is shown here. And on top of that, a large part of this information, of this non-coding information, is in fact coming from a series of repetitive DNA. And so you have nearly 50% of the human genome, so 1.6 gigabases, that are in fact not necessarily involved into the correct function. And you can imagine that perhaps we can shorten it at some point because it has not direct functional importance. We have to be cautious about that because well, you have also a lot of possible interaction, long distance interaction, and so sometimes you can get some paradoxical observation. But what is interesting is that this repetitive DNA is coming from different type of low complexity sequences. They can correspond to what we call interspersed repeats. So for instance, there are very characteristic ALU sequence, ALU elements, or lines, or DNA transposons, which are primarily derived from transposable elements. So probably it corresponds to the previous insertion of a virus into the genome, and then there is a sort of perpetuation with some molecules that allow the transfer of a copy at the second point in the genome. So this creates a series, a very large part of low complexity sequence that are all over the genome. And you have also other low complexity regions, such as the extremity of the chromosomes that are called the telomeres, the centromeres that are at the middle of the chromosomes. And what is also important is that you need to organize the chromosome, and you have to shape them to fit into the nuclei. And so there are different territories within the nucleus, and a lot of information is mediated by these different chromosomal territories. For instance, by interaction with the membrane, the nuclear lamina. So there is an interaction between the nuclear lamina and the chromatin. And this is generally well conserved, and you can arrive to pictures such as this one where you will resolve the different components, the different chromosomes in different entities that are interacting with the surface of the nuclei. So this is made possible through a lot and a very complex profile of interaction. You have, for instance, histones that will shape the nucleosome, that will shape the structure of the fiber of DNA, the different structuration of the chromosome until the creation of this full chromosome. And so the addition of hundreds and thousands of proteins is really crucial, and this is a very complex system to understand. But at the end, this is through this organization that you are able to put the 3 gigabase that represent, let's say, 1,000 copies of War and Peace from Tolstoy, to take an example of a big book. So you can imagine that in each copy of each nuclei with 5 micron diameter, you will have the storage of a big bookshelf that is possible. But so this is usually the type of idea that we are transmitting, to say the DNA storage can be particularly useful. But we need to consider that to do that, in the case of the mammalian system, what is needed is the very strict organization through this very elaborate system. And so this is perhaps something that needs to be more carefully thought for future application in the context of DNA storage. Yeah, so this is just an example of one of these jumping gene, the line retrotransposon. And so they can be inserted into the genome, they can be transcribed, they can go outside into the cytoplasm, they can reintegrate the genome, and they can reintegrate at a different place. And so this is how you can, and we have all of us have still some active line 1 copies that will represent up to 17% of the human genome. So not all line 1 are active, but a few of them. And so you can constantly have new modification with elements that are carried from one place to the other, and to have copy passed into your genome at different new locations. So yeah, so just to summarize, the big difference that I wanted to mention, and we can perhaps compare the viral genome versus the eukaryotic one. The level of compaction is extremely high in the 2 cases, because you can have a very high compression of the DNA molecules. But the mechanisms is through physical pressure in the case of the virus, while it is mediated by the histone wrapping and the hierarchical folding in the case of the eukaryotic genome. That makes particularly interesting the situation of the eukaryotic genome, because you are at low pressure and you regulate that by the state of chromatin. And behind that, you have other mechanisms of regulation, where you will eventually expose some genes at different positions into the nuclei to help their expression, for instance. So I have, I took that last slide just to mention So we are done with what I wanted to share about the organization of the genome. I think that what I wanted to share with you was the importance, the contextual, the importance of the context and the situations that you can have in the different models. And we have seen the difference that can exist between virus, between bacteria, between human cells. What I wanted also to share, and this is in line with the recent call that was launched by CNRS that Marc shared with me recently. So I don't know if it is the department, the life department or whatever, but they want to launch some new effort in terms of non-genomic contribution of biological mechanisms regarding information storage and transmission. And well, at some point, I would say that if you're able to store information in a network of neurons, you are doing this sort of thing. And so you have not only the genome by itself, you have not only the epigenetics that can carry information on long term, but you can also imagine that there are other aspects. And so, for instance, if you consider just a cell, through the specific organization of the cytoplasm, and that can be driven, for instance, by the substratum on which the cells has been adhering, you will create a very specific cytoskeleton, and so you will draw a specific pattern of differentiation. And so the information in biology is not 100% driven by the genetic information, and you have a lot of things that are driving possible interaction. We know all the chemical dialogue, the cellular networks, or just the physical properties of different constituents that can create concentration of gradients of some molecules inside the cytoplasm, or also affect the communication between cells by modifying the electrical activity of the membranes or things like that. And so there are probably a lot of other things that can be considered when you address the idea to store information through biology or through biological molecules. But I'm not sure that in the specific aspect of our project and for storing as nucleic acid, we are directly in line with something that can be proposed. That's it, and now I suggest there is a break. Yes? So we can have the break now if you want, and we can restart with only sequencing. Okay?